Hi, this is Philip Ador, founder of NCLEX RN 45-Day Challenge. In this video, I'm going to be talking about diabetes mellitus. In diabetes mellitus, your body has trouble moving glucose, which is the type of sugar from your blood into your cells. This leads to high levels of glucose in your blood and not enough it into your cells. And remember that your cells need glucose as a source of energy. So, not letting glucose enter means that the cells starve in the energy despite having glucose right on their doorsteps. In general, the body controls how much glucose is in the blood relative to how much gets into the cells with the two hormones, insulin and glucagon. Insulin is used to reduce blood glucose levels, and glucagon is used to increase blood glucose levels. Both of these hormones are produced by clusters of cells in the pancreas called islets of Langerhans. Insulin is secreted by beta cells in the center of these islets, and glucagon is secreted by the alpha cells in the periphery of the islets. Insulin reduces the amount of glucose in the blood by binding to the insulin receptors embedded into the cells membranes of the various insulin-responsive tissues, like muscle cells and adipose tissues. When activated, the insulin receptors cause vesicles containing glucose transporter that are inside the cells for fuse in the cell membrane, allowing glucose to be transported into the cell. Glucagon does exactly the opposite. It raises the blood glucose levels by getting the liver to generate new molecules of glucose from the other molecules, and also breakdown of glycogen into glucose so that it can all get dumped into the blood. Diabetes mellitus is diagnosed when blood glucose levels get too high, and this is seen among 10% of the world population. There are two types of diabetes, type 1 and type 2, and the main difference between them is the underlying mechanisms that causes the blood glucose levels to rise. About 10% of people with diabetes have type 1, and the remaining 90% of people with diabetes have type 2. Let's start with the type 1 diabetes mellitus, sometimes just called type 1 diabetes. In this situation, the body doesn't make enough insulin. The reason this happens is that in type 1 diabetes, there's type 4 hypersensitivity response or cell-mediated immune response, or a person's own T cells attack the pancreas. As a quick review, remember that the immune system that has T cells that react to all sorts of antigens, which are usually small peptides, polysaccharides, or lipids, and that some of these antigens are part of our own body cells. It doesn't make sense to allow the T cells that will attack our own cells to hang around, and so there's this process to eliminate them called self-tolerance. In type 1 diabetes, there's a genetic abnormality that causes a loss of self-tolerance among T cells that specifically target the beta cells antigens. Losing cell tolerance means that the T cells are allowed to recruit other immune cells and coordinate an attack on these beta cells. Losing beta cells means less insulin, and less insulin means that glucose piles up in the blood because it can't enter the body cell. One really important group of genes involved in regulations of the immune response is the human leukocyte antigen system, or the HLA system. Even though it's called a system, it's basically the group of genes on chromosome 6 that encode the major histocompatibility complex, or the MHC, which is a protein that's extremely important in helping the immune system recognize foreign molecules as well as maintaining self-tolerance. MHC is like the serving platter that the antigens are presented to the immune cells on. Interestingly, people with type 1 diabetes often have specific HLA genes in common with each other. One called HLA-DR3 and another called HLA-DR4, but this is just a genetic clue, right? Because not everyone with HLA-DR3 in HLA-DR4 develops diabetes. In diabetes mellitus type 1, destructions of beta cells usually starts early life, and sometimes up to 90% of the beta cells are destroyed before symptoms crop up. For clinical symptoms of uncontrolled diabetes, that all sound similar are polyphagia, glycosuria, polyuria, and polydipsia. Let's go through them one by one. Even though there's a lot of glucose in the blood, it cannot get into the cells, which leaves the cells starved for energy. So in response, adipose tissue starts breaking down fat called lipolysis, and muscle tissue starts breaking down proteins, but which results in weight loss for someone with uncontrolled diabetes. These catabolic states leave people feeling hungry, also known as the polyphagia, means eating, or poly means a lot. Now, with the high glucose levels, that means that when the blood gets filtered through the kidneys, some of it starts to spill into the urine, called glycosuria. Glycol refers to the glucose and urea, the urine. Since glucose is osmotically active, water tends to follow it, resulting in an increase in urinations or polyuria. Poly again refers to a lot, and urea again refers to urine. Finally, because there's much urinations, you will control uncontrolled diabetes because dehydrations and thirsty or polydipsia. Poly means a lot, and dipsia means thirst. Even though people with diabetes are not able to produce their own insulin, they can still respond to insulin, so treatment involves lifelong insulin therapy to regulate their blood glucose levels and basically enables their cells to use glucose. One really serious complication with diabetes is called diabetic ketoacidosis, or DKA. To understand it, let's go back to the process of lipolysis, or fat is broken down into the free fatty acids. After that happens, delivery turns to the fatty acids into the ketone bodies. Acetoacetic acid is a keto acid because it has a ketone group in the carboxylic acid group. Beta-hydroxybutyric acid, on the other hand, even though it's still one of the ketone bodies, isn't technically a keto acid. Since its ketone group has been reduced to a hydroxyl group, these ketone bodies are important because they can be used by cells for energy. But they also increase the acidity of the blood, which is why it's called ketoacidosis. And the blood becoming real acidic can have major effects throughout the body. Individuals can develop cosmal respirations, which is a deep, in-labored breathing as the body tries to move carbon dioxide out of the blood. In an effort to reduce its acidity, cells also have the transporter that exchanges hydrogen ions or protons for the potassium. When the blood gets acidic, it's by definition loaded with protons that get set into cells while potassium gets set into the fluid outside the cell. Insulin stimulates the sodium-potassium ATPases, which help the potassium into the cell. And so without insulin, more potassium stays in the fluid outside the cell. Both of these mechanisms lead to the increased potassium with fluid outside the cell, which quickly makes it into the blood and causes hyperkalemia. The potassium is then excreted, so over time, even though the blood potassium levels remain high, overall stores of potassium in the body, which include potassium inside the cells, starts to run low. Individuals will also have a high anion gap, which reflects a large difference in their unmeasured negative and positive ions in the serum, largely due to the buildup of keto acids. Diabetic ketoacidosis can happen even in the people who had already been diagnosed with diabetes and currently have some sort of insulin therapy. In states of stress like an infection, the body releases epinephrine, which in turn stimulates the release of glucagon. Too much glucagon can tip delicate hormonal balance of the glucagon and insulin in favor of elevating blood sugars and can lead into the cascades of even just described. Increased glucose in the blood, loss of glucose in the urine, loss of water, dehydrations, and in parallel need of the alternative energy generations of ketone bodies and ketoacidosis. Interestingly, both ketone bodies break down into acetone and escape as a gas by getting breathed out to the lungs, which gives a sweet, fruity smell to the person's breath. In general, though that's the only sweet thing about this illness, which also causes the nausea, vomiting, and even severe mental status changes, and an acute cerebral edema. Treatment of a DKA episodes involves giving plenty of fluids, which helps with the dehydration. Insulin, which helps lower blood glucose levels, and replacements of electrolytes like potassiums, all of which help to reverse the acidosis. Now, let's talk about type 2 diabetes, which is where the body makes insulin but the tissues don't respond as well to it. The exact reason why cells don't respond isn't fully understood. Essentially, the body is providing the normal amount of insulin, but the cells don't move their glucose transporter to their membranes in response, which remember is needed for the glucose to get into the cells. These cells therefore have insulin resistance. Some risk factors for insulin resistance are obesity, lack of exercise, and hypertension. The exact mechanisms are still being explored. For example, in excess of adipose tissue or fat is thought to cause the release of free fatty acids and the so-called adipokines, which are signaling molecules that can cause inflammation, which seems related to insulin resistance. However, many people that are obese are not diabetic. So genetic factors probably play a major role as well. We see this when we look at the twin studies as well. We're having a twin with type 2 diabetes increase the risk of developing type 2 diabetes, completely independently of the other environmental risk factors, and type 2 diabetes since tissues don't respond as well to normal levels of insulin. The body ends up producing the more insulin in order to get the same effect and move glucose out of the blood. They do the same through the beta cells, hyperplasia, and increased number of beta cells. And the beta cell hypertrophy where they actually grow in the size, all in an attempt to pump out more insulin. This works for a while, and by keeping insulin levels higher than the normal blood glucose levels, can be kept normal, called normal glycemia. Now, along with the insulin, beta cells secreted islet amyloid polypeptide, or amylate, so while beta cells are cranking out insulin, they also secrete an increased amount of amylin. Over time, amylin builds up and aggregates in the islets. These beta cells compensations, though it's not sustainable, and over time those maxed out beta cells get exhausted. And they become dysfunctional and undergo hypertrophy and get smaller as well as hypoplasia and die off. As beta cells are lost and insulin levels decrease, glucose levels in the blood start to increase and patients develop hyperglycemia, which leads to similar clinical signals that we mentioned before, like polyphagia, glycosuria, polyuria, and polydipsia. It means that the insulin-glucagon balance is such that diabetic ketoacidosis does not usually develop. Having said that, complications called hyperosmolar hyperglycemic state, or HHS, is much more common in type 2 diabetes than type 1 diabetes. And it causes increased plasma osmolarity due to the extreme dehydrations and concentrations of the blood. To help understand this, remember that the glucose is a polar molecule that cannot passively diffuse across cell membranes, which means that it acts as a solute. So, when the levels of glucose are super high in the blood, meaning its hyperosmolar state, the water starts to leave the body cells and enters into the blood vessels, leaving the cells relatively dry and shriveled rather than the plump and juicy. Blood vessels that are full of water led to the increased urinations and the total body dehydration. This is a very serious situation because the dehydrations of the body cells, and in particular to the brain, can cause a number of symptoms including mental status changes. In HHS, you can sometimes see the mild ketoanemia and acidosis, but not to the extent that it's seen in DKA. And in DKA, you can see some hyperosmolarity. So there's definitely overlap between these two syndromes. Besides, type 1 and type 2 diabetes are also a couple of other subtypes of diabetes mellitus. Gestational diabetes is when pregnant women have increased blood glucose, which is particularly during the third trimester. Although ultimately unknown, the cause is known to be related to pregnancy hormones that interfere with the insulin actions on the insulin receptors. Also, sometimes people can develop drug-induced diabetes, which is where medications have side effects that tend to increase blood glucose levels. The mechanisms for both of these is thought to be related to insulin resistance, like type 2 diabetes, rather than the autoimmune destructions process, like in type 1 diabetes. Diagnosing type 1 or type 2 diabetes is done by getting a sense of how much glucose floating around in the blood, and has a specific standards that the World Health Organization uses. Very commonly, as a fasting glucose test is taken where the person doesn't eat or drink, except for the water, for a total of 8 hours, and then has their blood tested for glucose levels. Levels of 100 milligrams per deciliter to 125 milligrams per deciliter indicates prediabetes, and 126 milligrams per deciliter or higher indicates diabetes. A non-fasting or random glucose test can be done at any time, with 200 milligrams per deciliter or higher being a red flag for diabetes. Another test is called an oral glucose tolerance test per person. Giving glucose and blood samples are taken at the time intervals to figure out how well it's being cleared from the blood. The most important interval being 2 hours later, levels of 140 milligrams per deciliter to 199 milligrams per deciliter indicates prediabetes, and 200 or above indicates diabetes. Another thing to know is that when the blood glucose levels get high, glucose can also stick up to the proteins that are floating around in the blood or in the cells. So that brings us to another type of test that can be done, which is the Hba1c test, which tests for the proportion of the hemoglobin in the red blood cells that has glucose stuffed to it and glycated hemoglobin. Hba1c levels of 5.7 to 6.4 indicate prediabetes, and 6.5 or higher indicates diabetes. These proportions of glycated hemoglobin doesn't change day to day, so it gives a sense of whether the blood glucose levels have been higher over the past 2 to 3 months. Finally, we have the C-Peptide Test, which tests the byproducts of the insulin productions. If the level of the C-peptides is low or absent, it means that the pancreas is no longer producing enough insulin and the glucose cannot enter to the cells. For type 1 diabetes, insulin is the only treatment option. For type 2 diabetes, on the other hand, lifestyle changes like weight loss and exchange along with the healthy diet and oral anti-diabetic medication, like metformin in several other classes, can sometimes be enough to reverse some of that insulin resistance and keep blood sugar levels in check. However, if oral anti-diabetic medications fail, type 2 diabetes can also be treated with insulin. Something to bear in mind is that insulin treatment comes with the risk of hypoglycemia, especially if the insulin is taken without a meal. Symptoms of hypoglycemia can be mild, like weakness, hunger, and shaking, but they can progress to a loss of consciousness in seizures and severe cases in mild cases. Drinking juices or eating candy or sugar might be enough to bring blood sugar up, but in severe cases, intravenous glucose should be given as soon as possible. The FDA has also recently approved intranasal glucagon as a treatment for severe hypoglycemia. Alright, now over time, high glucose levels can cause damage to the tiny blood vessels of the microvasculature. In arterioles, a process called Highline Arteriosclerosis is where the walls of the arterioles develop hyaline deposits, which are the deposits of the proteins, and this makes them hard and inflexible. In capillaries, the basement membrane can thicken and make it difficult for oxygens to easily move from the capillary to the tissues, causing hypoxemia. One of the most significant effects is that diabetes increases the risk of medium and large arterial wall damage and subsequent arteriosclerosis, which can lead to heart attacks and strokes, which are major causes of morbidity or mortality for patients with diabetes. In the eyes, diabetes can lead to retinopathy, And an evidence of that can be seen on the fundoscopic exam that shows cotton wool spots or flare of hemorrhages and can eventually cause blindness. In the kidneys, the afferent and the efferent arterioles, as well as the glomerulus itself, can get damaged, which can lead to a nephrotic syndrome that slowly diminishes the kidney's ability to filter blood over time and can ultimately lead to dialysis. Diabetes can also affect the functionings of nerve, causing symptoms like decrease in sensations in the toes and fingers, sometimes called the stocking glove distribution, as well as causes the autonomic nervous system to malfunction. The system controls a number of body functions, everything from sweating to passing gas. Finally, both the poor blood supply and nerve damage can lead to ulcers, typically on the feet, that don't heal quickly and can be pretty severe and need to be amputated. These are some of the complications of the uncontrolled diabetes, which is why it is important to diagnose and control diabetes through a healthy lifestyle. Medications to reduce the insulin resistance and even insulin therapy if beta cells have been Well, type 1 diabetes cannot be prevented and type 2 diabetes can. In fact, many people with diabetes can control their blood sugars levels really effectively and live a full and active life without any of the complications.